Good afternoon. Good afternoon, everyone. Thank you very much for joining our MHRC seminar series for the month of February. It's my pleasure to introduce Dr. Kristen Boyle, who's an associate professor from the Department of Pediatrics at Colorado University Anschutz Medical Campus. And Dr. Boyle is going to share with us today some exciting research that is highly regarded, as evidenced by her extensive NIH funding related to research that is focused on understanding how fetal exposures predispose infants to metabolic disease later in life. As you can see from the title, some very interesting models from stem cells to physiological models of muscle. And so with that, the presentation title focuses on mapping the earliest origins of metabolic disease in this lineage. So Kristen, thank you very much for joining us today. And I, for one, am certainly excited to watch this presentation.
Thank you so much for having me. I'm really excited to share this with you today.
Great. And I'll stop sharing here, and you're welcome to share your screen.
Okay. Okay. Thank you all for joining and inviting me to give this talk. First, I want to apologize for all the chaos going on down here lately. Hopefully that'll be resolved soon. And I also wanted to spend the first couple of slides talking a little bit about science communication. I came across this graphic the other day, and I think this may be something that we certainly here in the US are feeling about a mistrust and a mistrust in scientists. And I've been digging a lot into this lately, and so I just wanted to share a few tidbits that I've learned from this. This paper came out just last month in Nature Human Behavior, and it polled scientists across 68 countries in the globe, and it looked at trust in science and scientists. And we can see obviously North America, we have leaning where conservatives seem to have lower trust, and that's true for Europe as well. However, in Asia and some countries in Africa, we see that liberals may have lower trust. So I don't think that trust in science necessarily falls along political lines, but it does seem to be variable across different. But the most important thing I wanted to pull out from this work, they polled the role of science and scientists in policymaking across governments. And one of the most interesting facts here is that 83% of polled either agreed or strongly agreed that scientists should communicate about their science directly with the public. And so how do we do this? And I think when we talk about trust in science, I think we have to be careful in how we do this. And normally we communicate our science as a very linear method. We have a hypothesis, we do an experiment, we have results and a conclusion. But I found this book chapter here listed below that talks about science education and its role in our society in the development of democracy. And the issue they describe is that science actually happens more like this, where we backtrack, we question, our hypotheses are not true, we reevaluate how we're doing our science. We often present it as this linear method. And I think that presenting this as a polished narrative with clear authority does a disservice to how science is actually done and doesn't really adequately describe the nuance of science to the public. And in that sense, what that creates is this narrative that one study can be implemented into public health policy. And we know that's not true. It takes a body of work with a consensus coming together to create those sorts of things. And so I just want to keep that in mind as I'm presenting my research here today, because I'm going to present it this way, as the mess that it is, as the journey that I've done in developing this system. And I want to point out that this is not necessarily a departure from how I usually discuss my work. I really love the complexity of what I find. That's why I study all of my research in humans. The heterogeneity and the complexity of human disease is fascinating to me. And so I think we have to really embrace that when we're talking about our science. And in all honesty, it's when my hypotheses have not been true or have been slightly left of true that I really have found the most fascinating discoveries. So with that, I'll start my research talk for you here today. So my research focuses on the early origins of obesity and metabolic disease. We know that the gestational environment can impact offspring health. A lot of this work in the area focuses on obesity or gestational diabetes during pregnancy, and we know that those can increase nutrients and inflammatory or stress factors that are then impacting the placenta, that we also see similar patterns, and ultimately those can affect the developing fetus. And in those offspring exposed to some of these adverse nutritional environments during pregnancy, we see, elevated lipids, inflammation, similar as in their parent. We also see in humans, we can detect elevated fat mass at birth. And of course, from epidemiology and animal models, this increased risk of obesity and metabolic disease later in life. But let's dig in a little bit further here. I'm really interested in which children are at risk for metabolic disease. These are the latest CDC data that we have showing obesity rates, BMI above the 95th percentile in children. And we can see that, children over age 5 in the US, 20% or more have obesity. And this is relatively on par with Canadian children as well. But what's even more concerning is that the children in middle childhood that have obesity at the 6 to 11 year range, those children are most at risk for carrying that obesity into adulthood, along with serious such as heart disease, diabetes, and cancer. And I also want to point out, however, that there are many children, obviously, that do not develop obesity. So why is that? And who are these children that may be most at risk? This is a meta-analysis looking at obesity exposure in utero. So we see children age 1 to 14, the general population, about 14% of them develop obesity. However, in those that were exposed to obesity during gestation, we see quite a large, 2.5 times higher rate of obesity in those particular children. So yes, obesity exposure during pregnancy does contribute to risk of obesity later in life. But I want to show you this other meta-analysis here. This is looking at children with overweight or obesity, so a BMI percentile slightly lower. And if we look specifically at those exposed to normal weight during pregnancy, we still see that some of those children develop obesity by age 18. But in those that were exposed to obesity, we see half of them develop obesity by the age of 18. Obviously, this is concerning that half of these children are developing obesity. But what it also says is that not having obesity, these normal weight individuals, that does not necessarily protect us from developing obesity. Likely these are due to maybe more postnatal factors contributing to that obesity, but this increased risk here is concerning. Of those children exposed to obesity during gestation, there's this 50-50 chance. But what about these 50% that don't develop obesity? That heterogeneity is what really interests us. Why are these here in the half that don't develop obesity protected? Before I get too far, I do want to stress that while most of our research is in the context of obesity during gestation, we do consider this as a proxy measure for the many factors that contribute to and arise from obesity. So, all of these factors play a role in obesity development and the environment during gestation. And so while not all of these factors can directly affect their fetus, their impact on maternal metabolism and the milieu during gestation can impact the fetus. And so the concepts that I want to go over today for our talk are: 1, gestational exposures that contribute to offspring metabolic disease risk. In our lab exclusively a model of infant mesenchymal stem cells to inform pathways and the prediction of that obesity risk. And I'm going to obviously do a lot of work explaining to you what those are and how they work. But then where do we go from here? Looking towards optimal interventions to see if we can prevent childhood obesity. So what do we know about obesity during gestation? A lot of the pathways and the actual mechanisms that we understand really come from our animal models. Obviously, we have epidemiology in humans to back that up, but these mechanisms of tissue-specific effects are largely in animals. In adipose tissue, we see that these offspring have greater adiposity, larger adipocytes, adipose tissue inflammation. And in skeletal muscle, we see greater lipid deposition in the muscle, smaller muscle fibers, and altered metabolism, mainly lipid metabolism and oxidative metabolism. And so these can obviously appear very similar to what we see in adults with established obesity. And so we see these in the offspring as well. And the reason why these particular tissues are important is that they come from a common progenitor. During fetal development, the mesenchymal stem cell niche develops into all of the mesodermal tissues. Those mesodermal tissues are the adipocytes, osteocytes, myocytes, and chondrocytes. And as they develop into these tissues, they actually retain populations of those progenitor cells from the initial MSC niche. Is today as stromovascular cells or pre-adipocytes, bone marrow MSCs, skeletal muscle satellite cells, and synovial MSCs as well. And so these are the tissue resident progenitor cells that are there for the maintenance and repair of that tissue throughout our lifespan. So our model works under the premise that if there's a perturbation to the initial fetal stem cell niche, that can actually impact the development and maintenance of all of the mesodermal tissues throughout the lifespan for that child. And the great thing is that we can actually collect large amounts of infant MSCs from the umbilical cord tissue after the baby is born. We collect the tissue and culture out with tissue explants these same mesenchymal stem cells. And in them, we can detect these initial perturbations. And so mostly we use the model to study adipogenesis and myogenesis. I'm focused mostly on myogenesis here for my talk today, but our concept is that these initial stem cells that we get from the umbilical cord retain genetic and epigenetic signatures, and those epigenetic signatures are reflective of any programming effects from that intrauterine environment. We can then differentiate these cells in vitro to more mature tissue-specific phenotypes to understand how those initial genetic and epigenetic signatures inform the metabolic phenotypes of the differentiated cells. So our study design, we had the good fortune when we first started this investigation of piggybacking an epidemiological study here on campus. And so the parent study tracked 1,400 pregnant women in the station. We collected the MSCs in 164 of those infants. And then the epidemiological study has gone on to collect fat mass at several time points in the children. We've actually just finished collection at age 8 to 9, and children are now coming back for their 13 and 14-year visits. So in the future, hopefully we'll have some more longitudinal data on this. For most of the talk today, I'm really focusing on just those participants that we have stem cells for where the parents had gestational obesity. So those range between 15 to 20 women that had obesity. They met additional exclusion criteria as they had full-term infant eclampsia. So these are healthy obesity. They were frequency matched to normal weight pregnant women, and we matched on age, gestational age at delivery, infant sex, and the MSC proliferation rate measured by BRDU incorporation. We noticed differences obviously by design in pre-pregnancy BMI, in IR, triglycerides in the maternal circulation, and also greater adiposity in those infants at birth. And by nature of the two-compartment model, less percentage of fat-free mass. So our first study was really looking at this concept. The original slide I showed you in the sheep model of the fetal sheep where there's smaller muscle fibers and more lipid deposition, those authors put forth this hypothesis that was driven by this competition between adipogenesis and myogenesis during fetal development, that the mesenchymal stem cells were shifted more towards adipocytes as opposed to myocytes. So we wanted to look first at the extent of myogenesis in these cells. And so we measured several markers of myogenesis. We did not see overt differences in myogenin or myosin heavy chain across differentiation. But what we did find was that the cells from the obesity exposed infants had greater oil red, so this neutral lipid stain. And you can see that this red stain is within the muscle cells themselves. It's not necessarily lipid droplets or adipocyte spontaneous adipogenesis. It's really lipid deposition in the muscle cells. So we wanted to dig a little bit further into how that came to be. How are these cells accumulating lipid? Bear in mind, they have the same media, the same everything. They're treated the same once we collect those cells. And so at some point, they're just accumulating more lipid. And that can happen by more uptake or differences in lipid metabolism once it's in the cell. So we measured those components. And what we did was we used C14 labeled fatty acids, and we measured total uptake of fatty acids into the cell, which was not different. And then we measured esterification to triglycerides, which was higher in the OB-MSCs. And then we measured fatty acid oxidation, which was lower. Our fatty acid oxidation is a two-compartment model where we measure incomplete oxidation. We call that acid-soluble metabolites. Those are lipids that have entered the mitochondria and gone into beta oxidation, been completely oxidized to CO2 yet. And then obviously we have our complete oxidation to CO2. And note that this is all occurring before electron transport. So we kind of stop our measures at the TCA cycle. We saw that both acid-soluble metabolites and the complete oxidation, the incomplete and complete, were lower in the OB-MSCs. And this measure right here, this ASM over the CO2 ratio, is a fairly common index of the mitochondrial efficiency for fatty acid oxidation. So of the fats that get into the mitochondria, how many are completely oxidized? We did not see any overt differences, but this is where it gets a little bit messy. I then looked at the individual data, and honestly, I sat on this graph for over a year wondering what to do with this. We know from other studies that this higher levels of ASM CO2 ratio would indicate less metabolically healthy or less metabolically efficient. And I apologize, I'm in the process of changing my nomenclature in my slides. So sometimes it'll say healthy, sometimes it'll say efficient, but they're the same groups. So a higher ratio here would indicate less metabolically healthy or less efficiency for fatty acid oxidation, and then more here. And I sat on this for a long time until I decided to look at other variables in the children and in the mothers that may correspond to this. And sure enough, what we found was when we looked at maternal or gestational insulin levels, so circulating insulin levels during gestation, we see this stepwise increase with higher insulin in the less efficient group. Same thing with HOMA-IR and gestational free fatty acids. And in the cord blood, we see a very similar pattern. This is free fatty acids here are elevated in the less metabolically efficient group, and neonatal adiposity is also elevated. And so at this point, we started to believe that these were actually subtypes within our obese group. And it's also good to know that these data or similar results have actually been independently replicated by a group at East Carolina University, where they've shown that the acid-soluble metabolites are positively correlated with the infant fat mass at 1 month of age. And so we do think that this lipid metabolism phenotype is relevant to the infant and replicable. So the next thing that we did was to understand, what really is going on with metabolism here. We kind of came up with this hypothesis that the MSC phenotype that we're observing here is characterized for 1, the efficiency for fatty acid oxidation, and 2, the level of stored lipids. And that's kind of based on this fact that the normal weights have this huge spread of data here. And we're kind of wondering why. So we designed this experiment to test this a little bit further. And so this was our experimental design. We had our BSA control. We did excess fatty acid exposure for 24 hours, and this was an oleate palmitate lipid mix. And our hypothesis with this was that this would test whether lipid availability was a limiting factor in metabolism. And then we did that same condition with the addition of FCCP, a chemical uncoupler, during the fatty acid oxidation measure. And that was our test of whether metabolic rate was a limiting factor in our ability to oxidize these fats. And essentially what we found was that there was, quite a large increase in this fatty acid metabolism with our FCCP group. But you can clearly see that this metabolically inefficient group in the blue here just stays lower, and there is a significant interaction for this measure where this group here is lower and does not respond as robustly to the metabolic challenges that we're placing on it. So we do think that there is an intrinsic deficit in fatty acid oxidation in that blue metabolically unhealthy group. We went on to show, and here's just some summary of that. that in the normal weight, we see what we consider or describe as a more favorable metabolic milieu circulation in circulation during pregnancy, a robust response to metabolic challenge in vitro, and those children have low or relatively low fat. We see a slightly more favorable or metabolic milieu in one of the obese subgroups. We still see a robust response to metabolic challenge and lower fat mass in those infants. Whereas in that third group, we do see less favorable metabolic milieu, and this is all characterized by these three factors listed up above. We see an intrinsic deficit in fatty acid oxidation and elevated fat mass in those infants at birth. And so why is that? So we have actually done some epigenetic analyses in these cells, and these are done in the undifferentiated MSCs before we induce differentiation or test metabolism. And so we explored methylation in pathways specific to lipid metabolism, run all throughout here. And what we found was differential methylation on 4 genes. And you can see they're involved in the entry of fatty acids into the mitochondria, and then also one of the electron transport genes, SDHC. We see this is the increased methylation for all of these genes, and then some of these also have decreased or trend toward decreased mRNA abundance. And so we think these are actually affecting gene expression. And we're particularly interested in this PRKAG2 gene because that is a subunit for AMPK. And it was positively correlated with the adiposity of the infants at birth. And so let's dig a little bit deeper into AMPK. Here's an experiment we did with 24 hours of ICAR. So we have our MIM stands for a regular myogenous condition, and then we have our ICAR. And what you can see here is that we don't actually see much of a change or much of a difference in AMPK phosphorylation with the ICAR. But we see when we look downstream at the AMPK substrate ACC, we see this pattern here where we have, variations in ACC phosphorylation depending on these subgroups that we've identified, where the obese healthy group or more metabolically efficient group has the highest AMPK phosphorylation. But the thing to know about AMPK is that it's made up of 3 subunits. It has alpha, beta, and gamma subunits, each of which have 2 to 3 isoforms. And the specific configuration of those isoforms can affect the activity of AMPK. And here in this context, we're using this downstream marker ACC as a readout of the activity of the AMPK. And the point here is that the gamma subunit is the energy sensor of the cell, of the AMPK molecule. And so when there's ATP bound, that tells this AMPK that we don't need to make more energy, we have plenty. And so that's really more of an inhibitory signal. And there's also multiple phosphorylation sites that can affect activity. So AMPK phosphorylation on the alpha subunit can increase activation. And also AMP ADP binding on the gamma subunit, again, as the energy sensor, can regulate its activity as well. And there are other phosphorylation sites that can inhibit or activate AMPK respectively. So the thing that I want to point out here is that various combinations of all of these bindings of the phosphorylation sites or the ADP, AMP, things like that can affect the activity of AMPK. And when we look downstream, what we're really seeing is in the absence of huge increases in AMPK phosphorylation, we're seeing differences in the AMPK activity. And my thought is that if we have deficits in the gamma subunit, specifically the gamma 2 subunit, potentially that's disrupting the energy sensing component of AMPK. So one thing that we did is, knock down and overexpress this PRKAG2, the gamma 2 subunit. And so in the normal way, we used shRNA to decrease the expression, and we used in an obese cell, we used open reading frame to overexpress. And we repeated this experiment in these mutants that we've created. And what we can see here with our complete fatty acid oxidation, here's just the normal weight. This is our, control, our scrambled. And then here, when we knock down the PRKAG2, you can see it looks more like the less metabolically efficient blue group over here. And then when we do the opposite in the OBMSC, the scrambled, the control is down here, and that's what we see before, where it was the less metabolically efficient one. And when we overexpress, we've now created this where it looks more like the normal weight, the normal weight cell. And so we do think that PRKAG2 is important for this process of upregulating fatty acid oxidation and having this efficiency for fatty acid oxidation. But I will say a caveat, we've only been able to replicate this once. And so that's why we have not yet published it. We're looking to dig in a little bit more here and have some more repeatability on this. Our main issue is that the cells, the process of the transduction is quite stressful on the cells, and a lot of them die. So we want to be sure that we have good metabolically healthy cell data to look just in the presence or absence of this gamma-2. But what we have been doing is looking a little bit deeper into the lipid metabolism. And so our next goal was to do lipidomics in our myagenic MSCs. And so in this, I'm only going to show you parts of this paper. It was just published late last year. But we measured multiple lipid species, many lipid species in our myogenic differentiated MSCs. And we then just looked at the glycerolipids, the triacylglycerols and the diacylglycerols, and we did a k-means cluster, and that's a machine learning type approach where we ignore our initial phenotypes. We're going to ignore for now. We're putting all these normal weight and OBMSCs into the model, but we're going to ignore what we've identified as their phenotypes and let the machine learning process identify specific phenotypes regardless of what whether they're normal weight or OBMSC. And you can see kind of what we end up with here is 3 separate clusters. So they've said there's 3 phenotypes that are going on with these lipids that you've shown us. And these all are behaving similarly. And you can see there's some normal weight and some obese in each of the 3 clusters. And we hope that this is going to define more effectively what the phenotypes, the lipid metabolism phenotypes of these cells are, independent of our predefined notions of what normal weight and obesity mean during gestation. And you can see here, we've done a principal component analysis, and PC1 explains almost 80% of the variance, which shows us a very strong system here, a very strong phenotype that we're identifying. And you can see the differences in the clusters of the individuals. One thing I do want to point out, when you look at the loadings from the principal component analysis, you can see that PC1 is mainly driven by the triacylglycerols. So those triglyceride stores, and those appear to be elevated in cluster 2. And this kind of goes back with our hypothesis from one of the earlier papers, is that we think that the phenotype is characterized by the initial lipid stores that are in the cell and the ability to mobilize those lipids with a metabolic challenge. And that's kind of where we've been digging in all along. And so the rest of this paper does go on to show what happens with all of the various lipid species under various metabolic challenge conditions. And so if you want to take a look, you can look at those. But what I do want to show you here today is that we use these clusters that we've identified to look at what factors in gestation might be responsible. And what we find is we actually didn't find any overt relationship with insulin and glucose during pregnancy. But we did see a significant relationship with triglycerides, free fatty acids, HDL cholesterol, total cholesterol, and TNF alpha during gestation. And these measures were measured at 2 time points during gestation. And because all of these were related, we then decided to make a composite score. And the concept being that I think it's any one factor that may be solely responsible for what we're seeing in these cells. As you could see, there were 4 things that we measured there that were all related. And so we made a composite score, which is essentially a summed z-score of all of those measures I just showed you, including the glucose, which were not independently significant. And this is our modeling adjusted for maternal age, parity, pre-pregnancy BMI. So this is independent of maternal obesity, and also for gestational age at the blood draw. And we can see that that 3rd group here is much higher than the other 2 groups. At the offspring adiposity, we can very clearly see that that 3rd group, again, has the higher adiposity by age 5. And I do want to point out that this is not what we necessarily see at birth. At birth, we see that the orange group is lower and the purple and the green group are higher for adiposity at birth. But by age 5, we've been able to pinpoint and detect out that third group. I will say that because we started with about 37, 38 infants in this study, by the time we got out to age 5, it's a relatively small sample size of about 4 per group. So right now, one of our projects is we've actually measured the lipidomics in 70 children that we have age 5 adiposity for. And we're currently seeing if we can replicate these analyses across the 70 kids. And in that framework, we're actually doing this with lipid metabolism as well. So here we have 116 of the MSCs. So we're getting into larger, more epidemiological scale data now. And we differentiated ourselves to myocytes, and we looked at lipid metabolism. And these, as I showed you previously, in every single cell, we measured the lipid uptake, complete oxidation, and the ASM. And we actually did this in our control and our 24-hour fatty acid condition. And when we look at the principal component analysis, you can see that this explains about PC1 explains about 70% of the variance in the phenotype, and that there's relatively equal contribution of each of the measures that we made. And so in this context, because they seem to have a relatively equal impact on the phenotype, we then made another composite score where we called it our lipid metabolism composite score. And what we see, and again, that's a z-score of all of the measures summed together. What we can see is that the lipid metabolism down here on the y-axis, when we have higher lipid metabolism, those children have lower adiposity by age 5. And obviously lower lipid metabolism, they have higher adiposity by age 5. So we do think that the lipid metabolism of these cells is predictive of the adiposity of those children later in life. And we're doing this in various ways across cohorts. But we also want to know, if this is really informed by the original genetic or epigenetic signatures in the cells. And so we've now pulled back and we're looking at this model where we're actually we did RNA sequencing in our undifferentiated cells. So that's representative of the initial stem cell signature. And we looked for association with this MSC lipid metabolism score that we developed. And this is again in the 116 children. And we did did a pathway analysis, and essentially here's the summary of those data. We were looking for positive or negative associations between gene pathways and our lipid metabolism in the differential. And we're still in preparation in this paper and digging into the data a little bit, but two things that point out to me here is that we see pathways involved in mitochondrial translation are positively associated with our lipid metabolism differentiated cell. And the same thing, we see diseases of metabolism is negatively associated with our lipid metabolism in the cell, 21 days later after we've differentiated them. And so our point here is to help us to identify some potential targets in the undifferentiated cell that may be driving the metabolic phenotypes that we've just shown are linked to adiposity later in life. So to summarize so far, I think I've hoped I've convinced you even a little bit that maybe that multiple circulating factors during gestation might be contributing to offspring adiposity, though it does appear lipids may be most relevant in the context of gestational obesity. And also that stored lipids and the efficiency for fatty acid oxidation of the MSC myocytes is cumulative intrauterine exposure with mid-childhood adiposity. And all of those analyses, the associations that we did, were independent of maternal BMI. And I threw this in, I wasn't sure if we were going to have time, but I might take a few minutes with a body of work that we've looked at for the adipogenesis side of things as well. And so I might move a little quickly through this, but I just wanted to give a few little teasers of some of the fun things that we're doing on the adipogenesis side of this. We similarly looked at markers of adipogenesis across these groups that we had, and we do see that there's elevated markers of adipogenesis across 3 to 21 days. And similar to the myocytes, we saw increased oil red staining or increased lipids. And these are all markers of the adipogenesis. So we do think we have greater adipogenesis in the OBMSCs. And this was also correlated with adiposity of the infants at birth. One thing we've been questioning is whether greater adipogenesis may be a protective or not. We know as we look at adipogenesis, one of the key characteristics is adipocyte hypertrophy. And the extent of hypertrophy could potentially be metabolically detrimental. And when we look at the hyperplasia versus hypertrophy as two components of differentiation, excessive hypertrophy is often linked to inflammation or metabolic dysfunction. And so we really wanted to look at sizing and cell number in our model. So what we did was we developed a three-dimensional culture system so we could actually look at round lipid-filled adipocytes and size our cells from our MSC model. So we took our undifferentiated MSCs and we embedded them into a cross-linked collagen matrix, and then differentiated them to adipocytes for 14 days. And this is just one representative graph of what we found. We measured by confocal microscopy, and what we found was that, yes, there was larger cell sizes in the OBMSCs, the lipid droplets were larger, and it was actually shocking to us at the time. We didn't quite expect this because we embedded the exact same number of cells into every single bead, but by 14 days of adipogenesis, there were fewer cells in the obese group. We have a paper published on this now that digs in a little bit deeper into why that might be. But you can see here we did a bin cell sizing, and we do see across all of these cross-sectional areas of our cells that the normal weight do have more smaller cells, and the obese group has more larger cells. And on the epidemiological side, I call it but it's maybe mini epidemiological when we're talking about 100 or 150 to thousands. But we measured in 124 sets of cells our adipogenesis, and we looked at triglyceride content in the differentiated cells. Because we had seen before that this was related to the neonatal adiposity. And so we wanted to replicate this at larger scale. And what we found was, yes, the triglycerides are positively associated with infant adiposity. So this is 24 to 48 hours after birth. And we measured at 4 to 6 months, and we see it's also associated at that time point. And these are adjusted for a lot of common factors listed there below. But intriguing was that we also found that same association at 4 to 6 years. The triglycerides predict adiposity out that far. And I want to point out that this relationship here between the MSC triglyceride stores and the child adiposity was a stronger, more robust association than any other birth measure that we have. So usually we might use birth weight or birth adiposity as a predictor of long-term adiposity trajectories in those children. The MSCs were more robust. And I think even more intriguingly, they're also positively and quite strongly associated with child fasted glucose. So we do think that these MSCs can be a really nice predictor for long-term metabolic health consequences in the children. And so where do we go from here? Why are we doing this? I think one of the most important things is obviously we're interested in understanding the pathways that could be contributing to obesity development or metabolic disease development in these children. But really, at the end of the day, the reason we know that is because we want to maybe prevent or mitigate obesity in these children. And the reason I study, pregnancy and this fetal time point is really because that's the most optimal time for intervention. That's when epigenetic plasticity is highest. And if we can intervene in gestation or infancy, we have the biggest bang for our buck on reducing chronic disease risk across the lifespan. We may also want to consider preconception interventions, and we're actually writing a grant right now that will move into that space. And so one thing I just wanted to share with you as a little teaser, I have a grant under review Hopefully the review sessions are gonna continue, but I have a grant under review, supposed to be reviewed, that is looking at this at large scale. We actually have buy-in from several observational cohorts, large observational cohorts, and pregnancy intervention trials running around the country. We've actually trained multiple labs around the world now in how to collect and use these cells. And we're looking to basically combine all of these separate cohorts from intervention trials and observational studies, about 400 cells total, to see if we can do more digging into the gestational milieu and identify pertinent gestational exposures that are most predictive of the offspring MSCs that we're seeing. And we want to do that on two levels. One, what are the factors? We're going to do multi-omic analyses in maternal blood, and we're going to do this at different time points during gestation. So early gestation versus late. And the concept here is to really look for the ability to modify the gestational exposures that we think may be most relevant to the MSC outcomes. And in this, we're using these MSC outcomes as a proxy for the long-term metabolic health risks to the infant. And then when we do identify pertinent gestational exposures, what we would like to do is actually validate them in some, fancy 3D scaffolding models that we're working with a group in South Dakota who has some proprietary models that we would like to confirm our pertinent exposures in. And the idea is, if we can identify factors maybe early in gestation that we think are most predictive of offspring adiposity risk that we know are modifiable, we can intervene on those. So for example, say it's an inflammatory marker or inflammation in general, that may be something we can actually intervene on in pregnancy, because most of the pregnancy interventions to date focus on weight-based measures, maternal, or a general diagnosis of gestational diabetes as an intervention target, when we know it's the circulating, which is quite heterogeneous within those diagnoses. And so if we can intervene on specific targets like inflammation, as opposed to just weight loss in general, we may be more effective at mitigating the offspring adiposity risk. So to conclude, multiple circulating factors can contribute to the risk. And so we do think that more comprehensive milieu assessments are necessary to identify which factors might be most applicable. We think that the mesenchymal stem cells can inform precision prediction of obesity risk, both as outcomes based on intrauterine exposures, so we can use them in pregnancy intervention trials to look for outcomes, but also as predictive factors for future child adiposity. And our goal here is to really understand the most relevant gestational exposures for shaping offspring metabolic phenotypes, timing, and modifiability through these pregnancy trials. And so with that, I'd like to conclude and thank all the members of my team. We've worked with a lot of people who've gone on to do wonderful things, and obviously our funding sources, and a shout out to the wonderful research that NIH does fund, and I hope that they continue to fund.
Where am I? There I am. Okay. The background appeared, but I did not. Kristen, those slides are outstanding. I need you as a consultant on how to make slides of my own. And the research is really fascinating. One of the more translational talks I've seen in a long time. You know me, I have many, many questions, but my role here is to see if anyone else has questions. So please, everyone here, feel free to post questions in the chat. I'm happy to read it out. Or, and certainly preferred is if you just raise your hand, and I'll invite you to ask a question. Until I see some.
Do I leave the slides up or take them down, maybe?
Oh, you can leave them up, Kristen.
Okay.
No problem at all. Maybe I'll start with a question. I just keep bouncing between the biology, the applicability, and also the logistics of even trying to pull off these studies. And I don't know where to start. Maybe I'll start with the logistics. Like, I just want to verify with the study, particularly the one in JCI Insight with Gillenhammer as the first author. That was the same children from birth to 5 years of age,
yeah.
right?
Mm-hmm.
How? I've never heard of a study that long. That must have been difficult to pull
Well,
off.
so if you caught it at the beginning, I got this idea to do this after my first child was born. And I was bumbling around trying to get some chords. And I just had the good fortune. It was like this serendipitous. I talked to a graduate student. She's like, my mentor, we had this epidemiology study and they were collecting cord blood at the time. And she actually was really excited about the model, the graduate student was. And so she convinced her PI that we needed to start collecting the MSCs. Originally doing this, I was a postdoc, just kind of starting to get my own work for my own lab. And I was just going to collect like 12 normal weight and 12 obesity and try to see what I could find. And she came along and we started collecting them, and we're just like collecting as many as we can. And we ended up with 160 some odd cells, and the epidemiology studies, completely separately funded. And so that just runs in the background, and I get the benefit of just mining the data as it comes in. And so these kids are now, actually my two kids are in the study. I only have my son, my youngest. And actually those adipocytes that were twirling around, those are,
Okay.
but, so the kids are like 13 now.
Right.
Mm-hmm.
Right.
And we keep getting data on all the kids. Yeah.
Oh yeah, it's still ongoing.
Oh
Okay.
yeah.
That's amazing.
Yeah.
That's amazing. Mary Ellen, please go right ahead, and thank you for coming.
Thanks, Chris. There we go. Thanks very much for that great presentation, Kristen. I really
Thank you.
enjoyed it. Exciting and really important research. So I have a few questions like Chris. This is really, really exciting, but I'll try to communicate the two that I have right now. So one is about, the acid soluble fraction, the acid soluble metabolites, and efficient and inefficient fatty acid oxidation. So if the acid soluble metabolites in the less healthy, less efficient fat burners, if the proportion is up. So do you have an idea where those carbon units go? Have people done tracing to see, which, like, where those metabolites go? Do they traditionally, are they, within the TCA cycle, or have they gone off into, other redox pathways? Do we know?
Right. I don't know that anyone's done tracers to tell exactly where they go, but that is the assumption that they're either, somewhere in the middle. And if there's like, say, a deficit in beta oxidation, for example, they might be stuck in the middle there, but that's where those, peroxidized lipids can then come from, because they're kind of just sitting there around like halfway through the cycle. And the thing about the acid soluble metabolites is we don't know, like, it's a measure of anything that where the BSA has been cleaved. And so the concept is that they've entered the mitochondria, they're just not CO2. And aside from that, with the measure that we do, we don't exactly know. But it's a really interesting question. But I do think if there's an abundance of them, they would be and could some damage that way.
Yeah. That's really cool. So my other question was related to your decreased lipid oxidation and the predictive nature of that in adiposity in the children. And so that's consistent, with the overall conclusion that, energy expenditure beyond energy intake in terms of the development of obesity, that energy expenditure itself is very important.
Right.
But could these data still be interpreted that the fatty acid oxidation is higher because there is a lower energy intake? And so therefore, if there's a lower energy intake, you have higher, fatty acid oxidation. So is it the chicken or the egg, or is it, the cart or the horse? So what is driving this, and what does it mean?
I don't think that we have the answer. That may be something we could actually look into, because we do have dietary data on the children. What we did, though, in all of those analyses, we did adjust. We have 7-day actigraph data on those children as well.
Yeah.
And we have ASA 24 data for the dietary on the children.
Yeah.
We did use those measures and we adjusted for them in those predictive models. But we use them as, we kind of made the the total energy expenditure over I forget exactly which variable we used, but we, adjusted for the physical activity, and then we used healthy eating index as the adjuster for the diet, but we haven't looked specifically at caloric intake on that. So we kind of semi-adjusted for some of those factors, but maybe not the way you're talking about.
Yeah, wow, lots of great data. Yeah, thanks very much again.
Thank you.
Thank you, Mary Ellen. Great conversation there. We've got 4 questions in the chat group, so I'll read them out here. Let's see. One is from Orlando Sudia here at York. Were there differences in physical activity levels among the pregnant women who donated the MSCs? And if so, did it affect the metabolic phenotype or the epigenetic profiles?
So the only information that we have on physical activity in gestation is through self-report. So unfortunately, we don't have like actual direct. And I'm going to say the data are messy. And so we have tried to dig into that, but haven't found any overt patterns that would indicate that the physical activity is, influencing the MSC phenotype. However, Linda May and Joe Homar, East Carolina University, they have run a pregnancy intervention trial, and they've started collecting the MSCs from that trial. And what they show, they have a couple papers out now. They do show that the exercise intervention in pregnancy does impact the MSC myogenesis lipid metabolism. So exactly what we've shown is important here. And we're actually working in collaboration with them to dig a little bit deeper on that. So Nick Broski, who's an assistant professor there, has an R01 that's really going to look at the mitochondrial function in the cells from those infants. And we're going to do a multi-omic, genetic, epigenetic RNA sequencing analysis to really define exposure versus genetic influences and how the intervention, the exercise intervention, really impacts the stem cell phenotypes.
Okay.
So in the works.
You have a lot of good collaborations on the way
Yeah.
go here. That's great. Okay, we've got again, anyone else that wants to raise their hand, feel free to do so. But I've got 3 questions that was sent to me direct message from Anthony Shime, who's also here at York. The first one is, are the MSCs you study the same mesodermal cell types that give rise to the embryonic and fetal myoblasts during gestational myogenesis?
Yes. I mean, so for context, we're collecting them from the umbilical cord. So these were never in the fetus per se, but these niches are developed, before the cord and the placenta are developed. So essentially, if you consider them like a hierarchy, the mesenchymal stem cell niche developed in and the cord is developed in the epiblast, the blastocyst, and it kind of just branches off. And some of those go to the cord tissue where we're collecting them, and some of them go to the fetus, where then, yes, they would be the progenitors for those cell types that you're talking about. So we kind of think of it as they're from the same originating niche, but they've never been, we call them tissue naive. They've never been developed in the adipose or the muscle tissue of that infant in the fetus, or of the fetus during development.
Okay. Okay. Right. Okay. Thank you. Anthony Shime's second question: do you sort the cells when isolating, or are they a heterogeneous population?
They are a heterogeneous population. We do not sort them, and we do that on purpose because we think, and we haven't had funding yet, we would love to do single cell RNA sequencing on the original population to see what that heterogeneity is, but we don't have funding. And the reason why is because tissues are heterogeneous, and we consider heterogeneous population as what's going to be developing those fetal tissues. So that's why we want to keep that there. And the cool thing about the cells, the mesodermal cells, characteristics is that they're plastic adherent. So essentially, we just put the tissue pieces down on the plastic, and they just grow out. And all of the other cell types that might be in the cord tissue are non-plastic adherent. We do characterize them for purity. So we do some flow characterization afterwards to make sure, yes, they are 98 to 99% positive for MSC populations, but they're a heterogeneous one.
Okay, very clear. And his next question was, do you see differences in, oops, sorry, it moved. Do you see differences in male and female offspring?
Great question. We've been trying to find, and we thought we had one, but it didn't pan out. Even on large scale on the 125, we've looked for any sex differences in lipid metabolism. We've looked for sex differences in our RNA sequencing. We've looked, and we have not identified any as of yet. In our smaller groups where we're looking at 10 or 20 per group, we're definitely not seeing any sex differences. And it is a really interesting concept. And it makes you wonder how much is, hormones with tissue development, and how, tissues may develop differently in a male or a female fetus, or even, later in life postnatally, that develops differences in metabolism. But yeah, we're looking and we're not finding any.
Okay. Great detail. Anthony just messaged me. Please thank her for all these great answers.
Thank you.
All right. We've got a minute to go. Anyone else have a question? Any students? Don't be nervous. Okay. All right. Well, I won't use up if I ask one question with 1 minute left, it's not going to be 1 minute. So I'll have to talk to you another time. But Kristen, thank you so much for this really engaging talk, fascinating work. And as you saw, enormous interest here. And everyone else, please join me in thanking Kristen Boyle for this excellent presentation. Thank you so much.
Thank you so much.
And on that note, I'd like to thank you all as well for joining us for our seminar today. Please keep an eye out for emails if you're on our email list, or our social media accounts at the Muscle Health Research Center on LinkedIn or Twitter, or my own account for our next seminar, which will be in March. And on that note, everything's finished. Have a great weekend. And Kristen, if you could